So here I am going to start another new chapter, that is economic importance of bacteria and fungi. In this video you will mainly read about economic importance of bacteria, and on the next video I will teach you the economic importance of fungi. So you know what is bacteria? You have already learned earlier about it, that bacteria are primitive organisms. They are prokaryotic, means they do not have well-defined nucleus. They are unicellular organisms, and each bacterial cell contains a single chromosome, that is nuclear material, which is not enclosed in a nuclear membrane. That is why they are called prokaryotic. So they are the smallest living creature on earth, whom we cannot see without microscope. They are so numerous in number and can be found everywhere, whether it is air, water, soil, foodstuff, or inside our body. So there are thousands of bacteria in our mouth, on skin, inside intestine, in the genital tract, etc. Certain bacteria are highly injurious, causing diseases. But there are others as well which are highly beneficial because they can synthesize vitamins such as vitamin B. Their size can be of 2 micrometer long and 0.5 micrometer thick. Bacteria are found in many shapes. For example, cocci means spherical bacteria, as you can see in the figure 11.1. Bacilli, that is rod shape like structure. Next is spirilla, means spiral or twisted bacteria. And the next one is vibrio, means comma shaped bacteria. As we know, bacteria are unicellular, so they live single, but some occur in pair as well. So in coccus you can see they are found as diplococci, that is in pair, or in long chain, that is streptococci, or in clusters, that is staphylococci. Now let's read about the structure of bacteria. Bacteria contain the living substance called protoplasm. Cytoplasm, nucleus, and other cell organelles form protoplasm, which is surrounded by a non-living stiff cell wall. In bacteria, cell wall is made up of peptidoglycan. It is a kind of chemical which consists of some sugars and amino acids. Cytoplasm is surrounded by a thin cell membrane, which is further surrounded by cell wall. As we know, bacteria do not have well-defined nucleus, but chromatin, that is DNA material, is present in the central region, which is in the form of single circle and is not enclosed in a nuclear membrane as we see in eukaryotic cells. This chromatin material is attached to the cell membrane at some point. Some bacteria have slimy protective layer called capsule. It is the outermost layer of the bacterial cell. Capsule protect the bacteria from desiccation, and it is also responsible for causing diseases. Most bacteria cannot move by their own. They are only passively transported by wind, water, or contact. Some bacteria possess one or more whip-like flagella, which provide active locomotion in liquid environment. Most of the bacteria are heterotrophic in nature, means they depend on other for food. They can be either saprotrophic or parasitic. Some bacteria are aerobic, means they require oxygen for respiration, whereas some bacteria do not require oxygen and are called anaerobic bacteria. Bacteria shows asexual reproduction by fission or cell division. In figure 11.3, you can see a bacterial cell where the circular DNA duplicates first. Then the cell expands and the two DNA are pulled apart. The cell constricts at the middle, separating the two cells. There are very few bacteria which show highly primitive type of sexual reproduction, which is also called conjugation. In conjugation, two bacteria of different strains but of the same species come together, and the plasmid of the donor may be transferred into the recipient through a hollow tube like extension of the donor cell. Some bacteria have the tendency to overcome unfavorable conditions by forming spore. When the conditions are unfavorable, the bacterial cell draws its content into a spherical mass, which becomes surrounded by a thick and hard protective wall. And this little round body is a spore, which is contained within the original cell wall, as you can see in figure 11.4. These spores can tolerate extreme dryness. Sometimes even they cannot be killed at high temperature of boiling water or at very low temperature of frozen ice. They can even tolerate poisonous chemicals. They remain as it is and do not divide until the favorable condition arrives. On meeting favorable condition, these spores germinate. Their dormant protoplasm becomes active again and stream out of the ruptured wall in the form of a new bacterium. So in this manner, this invisible creature escaped the unfavorable condition. Bacteria are used in medicine in the production of antibiotics, serum and vaccines. So what is antibiotic? It is a chemical substance produced by a living organism which can stop the growth of some disease-producing bacteria and fungi. There are different types of antibiotics used for numerous infectious diseases. So, the first produced antibiotic is penicillin, which was derived from a fungus, not bacterium. It was first observed by Alexander Fleming when some bacteria were grown in a culture and were destroyed accidentally through contamination of a mold Penicillium notatum. Then he discovered that this mold produced a substance which suppressed the growth of other bacteria, and the substance was named penicillin. In this manner, several antibiotics were discovered, which are today well known to us. There are some criteria for a good antibiotic. Means the antibiotic should be able to kill a variety of disease-producing microorganisms, which is also called broad-spectrum antibiotic. There are certain good bacteria also in our intestine, so it should not kill them, and it should not produce undesirable side effects. Next is serum. Suppose if I remove the fibrinogen from the blood, then the plasma which remains there is called serum. This serum contains numerous chemical substances including antitoxin. Antitoxin is a substance produced in animal's body which react with the poison or toxin produced by the invading germs. Now let's see how this serum is actually prepared. First, a small dose of bacterial toxin is introduced into the blood of a healthy animal, for example, horse or cow. Then the body started producing antitoxin against it to neutralize the effect of toxin. After several injections of the same toxin, the blood of such animal is taken out. On chilling, it clears as straw-colored fluid, which separate out from the clot, and the straw-colored fluid or liquid is called serum. There is a bacteria named Escherichia coli or E. coli grows very rapidly. So through genetic engineering, the particular gene-dependent products are extracted from these bacteria. For example, the hormone insulin, which was the first such substance produced by E. coli. Next is vaccines. As we know, vaccines are available for a variety of diseases such as cholera, typhoid, chickenpox, etc. A vaccine consists of weakened or dead germ substances which are introduced into the body for developing resistance to a particular disease. Germs are injected in such a low concentration that it can form mild disease or no disease. but the body gets stimulated to produce antibodies or antitoxins against that particular disease. TB vaccine is used for typhoid. TB means typhoid, paratyphoid A and paratyphoid B, which consists of kill bacteria. BCG vaccine, that is Bacillus calmette-guerin, is used for tuberculosis. This vaccine consists of living weakened bacteria. Toxoid are chemically modified toxin, or we can say inactivated toxin of the particular bacteria, which is no longer toxic but can be used for the production of antibodies against a particular disease, such as those for producing immunity against diphtheria and tetanus.